Imagine facing a future where your once steady balance gradually slips away, leaving you with stumbling steps. And when you try to communicate, slurred speech makes it hard for others to understand your words. Even the simplest tasks that were once effortless now presents a daunting challenge. As the condition worsens, you find yourself bedridden, unable to take care of yourself. Well, these are what patients with different types of spinocerebellar ataxias, SCAs, and Huntington's disease experience. These conditions share a common thread: a genetic mutation with CUHK trinucleotide repeat expansions in affected genes, which is transcribed and translated into toxic polyglutamine, or polyQ domain-containing proteins. That is why the diseases we just mentioned are also known as polyQ diseases. Unfortunately, to date, polyQ diseases remain incurable. But how does this genetic mutation actually cause the neurodegenerative polyQ disorders? To understand this, let's take a closer look into the nucleolus. The nucleolus is the largest structure in the nucleus of eukaryotic cells, which is responsible for the synthesis of ribosomes that reads the messenger RNA sequence and translates that genetic code into amino acid sequences in the cytoplasm. Under normal conditions, human nuclear proteins, nucleolins, binds to the upstream control elements, and rRNA promoter initiates pre-rRNA transcription and regulates ribosome biogenesis. However, in polyglutamine diseases, expanded CUHK RNA disrupts this interaction by recruiting NCL in the nucleus, leading to the downregulation of pre-rRNA transcription and the failure in ribosome biogenesis. In the process, neurotoxic species such as small CUHK RNA and CUHK RNA foci will also be formed. These disruptive events eventually triggers neuronal apoptosis and neurodegeneration, contributing to the devastating symptoms of these diseases. But here comes a potential game changer. In 2016, Professor Chan collaborated with Professor Ngo from the School of Life Sciences to develop a 13 amino acid peptide inhibitor, P3, against CUHK RNA-induced nuclear stress in polyQ diseases, which was further developed into a more structurally stable 21 amino acid peptide inhibitor in 2018. This peptide, called the beta-structured inhibitor for neurodegenerative diseases, BIND, works by containing the sequence of the ribonucleoprotein domain motif of NCL RNA recognition motif 2, which allows it to mimic NCL and be captured by expanded CUHK RNAs, so that NCL can be released back to resume its normal functions in the nucleolus. By capturing expanded CUHK RNAs, BIND effectively reduced downstream neurotoxic species, including small CUHK RNAs, CUHK RNA foci, and polyQ protein aggregates in vitro. This reduction in neurotoxic species resulted in a suppression of neurodegeneration and improved cellular functions. BIND treatment also showed promising results in vivo using a Drosophila model of polyglutamine diseases. It ameliorated retinal degeneration, improved climbing defects, and significantly expanded the lifespan of Drosophila expressing expanded CUHK RNAs. Surprisingly, Professor Chan's research team further discovered that BIND can also be used for treating amyotrophic lateral sclerosis, ALS, which is characterized by gradual degeneration of nerve cells in both the brain and the spinal cord, leading to muscle spasms, slurred speech, and difficulty in swallowing and breathing. In brief, instead of a genetic mutation with CUHK trinucleotide repeat expansions as in polyQ diseases, ALS is associated with GGGGCC repeat expansions in affected genes. And RNAs transcribed from the expansions can form toxic RNA species, including RNA foci, as well as toxic proteins known as dipeptide repeat proteins, DPRs, through translation. However, by direct interaction with expanded GGGGCC repeat RNAs, BIND can suppress the formation of toxic RNA foci and DPRs in vitro, opening up a potential therapeutic treatment for ALS. Since 1999, Professor Chan has been studying polyQ diseases and developing therapeutic interventions against these diseases. In fact, to push forward drug development projects and provide patient care services, Professor Chen founded a technology startup company named Rare Power in 2023. To learn more about the company and its work, you may visit their website and profiles on LinkedIn, Facebook, or Instagram. The development of BIND opens possibilities for potential therapeutic approaches in combating the toxicity induced by expanded CAG RNAs in polyQ diseases. Hopefully, in the near future, BIND can be further developed into therapeutic usage and bring hope to those affected.